Now, last time I have explained you about the organ culture. So organ culture is a type of tissue culture that's involve isolating an organ for in vitro growth. Now, what different types of organ you can culture? Shoot, root, leaf, and flower. So this method is used for preserve the structure or function which allows the organ to still resemble and retain characteristics they would have in vitro. Means the function main function is to allow this organ to perform its function and have its morphology as good as they are functioning in the natural environment. Then here, new growth continues given the organ retains its physiological features. Means the growth of organ continues. For example, if you are doing root, the growth of root will go on. If you are doing shoot, the shoots will develop new leaves. It will increase in height, width. So here there is no compromise with its function. And as such, organ health provide information on pattern of different growth, differentiation as well as development. Now significance, experimental system for study nutrient and growth factor. Last time I explained you. Then study the interdependence of organs for growth hormones and their other growth factors. Then study of morphogenesis and biosynthesis of specific metabolites and new avenues in development in agriculture and horticulture. Now this slide also I've explained you, that is organ culture. It is divided into two types: vegetative organ culture and reproductive. In vegetative comes root, leaf, and shoot tip culture, and in reproductive comes flower, isolated ovary, and ovule or embryo culture. Hello.
हो सर. हॅलो.
Ah. So what is mean by leaf culture? Now the leaf culture is culture of excised young leaf primordia or immature young leaf of the shoot apex in a chemically defined medium where they grow and follow the developmental sequence under control conditions. So we'll cut this sentence into different parts. So leaf culture is culture of excised young leaf primordia. Now this is
प्रायमोरिडियम म्हणजे काय सर?
primordia means tip of the leaf. Why they are taking tip of the leaf? Can anyone tell me? Or immature leaf
हाय
only? Why?
हाय ग्रोथ रेट असतं ना तिथे.
Ah, due to what? कशामुळे हाय ग्रोथ असणार तिथे?
द प्रेझेन्स ऑफ मोर जिब्रेलिन्स.
No, due to presence of meristematic tissue. बरोबर ना? The main aim of
हां.
meristematic tissue is continuous development.
येस सर.
We can
कंटिन्युअस
develop
सेल
this.
डिव्हिजन.
We can develop cultures from also non-meristematic tissue, but growth rate here is very less. So we are going to focus on high growth rate. Now where it is seen, shoot tip, root tip, leaf primordia, and immature leaves.
येस सर.
Then it is grown in a chemically defined medium. Why chemically defined medium? Can only leaf जर आपण आणलं आणि if we put this leaf in only just simple water, can it grow? No. असं तर whatever vegetables we are bringing, coriander, methi, palak, chepu, or these vegetables, they would have grown easily.
येस
Here
सर.
we have to give chemically defined media. Now chemically defined media means it should have all 16 microelements, macroelements, growth hormones, and aseptic conditions. Barabar, this is middle part.
येस सर.
Where they grow and follow the developmental sequence under control conditions. Now control conditions is if you give media, everything you give, and you put this culture in 50 degrees Celsius, what will happen? They will get died because their activity is maximum at 28 degrees Celsius. If you are putting all this media, everything, and you are keeping this inoculated test tubes or petiplex in a dark place, no light is given, there will be no development of shoot. So control conditions are very important. Then pH, temperature, pH, light, all these are control conditions.
Yes, sir.
Now, most of the work of this leaf culture has been done with lower plants. Now, earlier this was done with fern. Fern is a pteridophyte. Name example is Osmunda. Although higher plant species such as tobacco and sunflower have been used, major work is done in lower plants. In culture, the fern leaf primordia 1 to 2 mm excised from underground buds developed into leaves having a normal morphology except that they are much reduced in size than in vivo leaves due to reduced number of cells rather than decrease in cell size. Now what they did, uh, in our botanical garden we have got fern. There is a jackfruit tree. On that jackfruit tree we have got lot of collection of ferns. So how they develop this leaf primordia fern, they keep on unrolling themselves and they will grow in a whole leaf. So what they did, scientists, they excised this leaf primordia and put it into culture medium. Here the growth was normal, but only the size was reduced as compared to the natural plant size of the leaves. Now, growth of the culture leaf primordia is also completed earlier than intact leaf. If you consider the growth was completed earlier because everything is optimum. Optimum chemicals, optimum environmental conditions. In all these optimum things, the growth is very healthy. But in nature, what happens? There will be some heat stress is there, water stress is there, environmental factors are there, temperature will increase, in night it will fall. So growth in culture is very high. Now principle, the leaf primordia or very young leaves are excised, surface sterilized and inoculated on the agar solidified medium. First step is selection of explant. Explant selection झाल्यानंतर काय करतो आपण? What do we do? It's sterilization. For surface sterilization, what we'll use now? What?
Alcohol based. Alcohol.
Alcohol. How much percentage?
75%.
60 to 70%. Now, whatever sanitizers we are getting in the market, they also should contain at least above 60 or 70% ethanol. Then only they will be effective. Alcohol. Now, here if you are using alcohol, alcohol there are a lot of different types. Example are methanol, ethanol, propanol, butanol. There are a number of types of alcohol. In this, which alcohol we use? Can anyone tell? In tissue culture, which alcohol we use?
Ethanol.
Ethanol. Yes, exactly. Why they are using ethanol? Whatever alcohol beverages are present, whatever alcohol beverages, examples are wine, rum, vodka, whiskey, all those things, they contain ethanol. Means if we consume it by chance, there is no any damage. Also, if we rub this alcohol on our body, there is also no problem. Ethanol. But for example, ethanol formula is ch2 ch3 sorry c2h5oh ethanol, and methanol is ch3oh. Only one ch2 is extra if you consider structure. But methanol, if you consume methanol, it is poisonous. Person can die due to overconsumption of this methanol. See, only one CH3 molecule was added and the property has changed. So they use ethanol. They will not use other alcohol. Sometimes they will use isopropanol because it is also less toxic compared to methanol. Alakshat, so you should remember which alcohol they are using. Hello?
Hello, sir.
Ah. So which alcohol they are using? Ethanol, and ethanol is also produced by yeast fermentation.
Yes, sir.
Then next process is to be cultivated on a solidified medium. So why solidified medium? Can anyone tell? Why not liquid medium? Everyone has known the question what I've asked. Why they are using solid media only and not liquid media? Can anyone tell?
So solid media contains more amount of nutrients as compared to
It
liquid
is
media.
not at all like that, because we are preparing all our media in liquid form only, and you are only adding agar agar to solidify.
Agar agar.
So nutrient is not this factor. Imagine you have taken 20 ml of liquid culture in a test tube, and you are inoculating a leaf. What will happen? The leaf is either heavy, it will sink to the bottom, or it is light, it will float. Can it be holded properly?
No.
No. But here in solid media, what you can do? 30% of that leaf you can put in, immerse it into solid culture, and remaining 70% can be in the atmosphere. So what will be? It will be useful for to respiration of that leaf. It can be done respiration. Alakshat. So here we use solid media. But if you use liquid media, if it is synced at the bottom, what will happen? There will be no proper respiration. Plants are not amphibians. Some plants are, but all plants are not amphibians.
Yes, sir.
Then leaves can be taken from aseptically grown plants for culture. Now see, when you are selecting a seed, what happens? Embryo is inside the seed. If you are taking shoot are inside the many covers of the leaf. But here you are taking leaf, then on that leaf surface lot of bacteria, fungi are there. So here it is advised, what it is advised, that is you have to take a leaf which is grown aseptically. The plant is cultured, and from that plant means, for example, you will take the seed, you will grow that seed, you will sterilize that seed, and you will grow that seed aseptically in a conical flask. Now the whatever the shoots and leaves it will produce, it they are also aseptic. From that you will excise a leaf and take it for your experiment. Last is leaf primordia are very young leaf, have more growth potential than nearly mature leaves. I've explained this. Why leaf primordia? They are young. So as they are young, they will contain more meristematic tissue. And mature leaves, after maturity, leaves get senescence. Senescence means they will kill themselves. And in whatever waste leaves are fallen on the ground, they are senescence leaf. So it is of no use. See here you can see the diagram. Healthy plant, from that they have excised a leaf. From that leaf in figure C you will get callus. Here in leaf culture you will not go for callus directly, you will get from that plants regenerated, and then you will plant it. Now here in this figure you can see a figure. Can you see how these shoots are arise from that plant leaf? Can everyone see? A figure मध्ये दिसलं का सगळ्यांना?
हो सर.
Yes, sir.
Then B मध्ये काय झालं? These whatever the leaf primordial shoots they have there, they are been separated and put separate in the culture. Then what happens? The shoot grows. In figure C, what happens? They have put that culture into rooting media. White color root you can see. In D, what happens? There is shoot, there is some stem part, and downwards there is root. So they will remove this from the culture and put it into soil for hardening. Got it?
Yes, sir.
Now see, this is the example of leaf. Directly they have taken leaf, and from that leaf they have got many shoots. So I will tell you example of tobacco. Tobacco is a model plant used for transformation, genetic transformation. Why it is used? Can anyone tell? Can anyone tell? क्वेश्चन कळालं सगळ्यांना? Tobacco is a modern plant for transformation.
Sir, for alcohol production, more amount.
अल्कोहोल तर टोबॅको पासून प्रोड्यूस करतच नाही. टोबॅको पासून काय प्रोड्यूस करतात?
Uh,
Whatever tobacco they are used for chewing or cigarettes, it is produced from tobacco. Alcohol nahi. Alcohol we are producing from sugar cane because it has got sugar content. Tobacco it has got nicotine. First of all, transformation, agrobacterian transformation is easy in dicotyledon plants. So tobacco is a dicot plant. Now if we consider tobacco where it is grown in tropical regions. Now what are tropical countries? India is a tropical country. Temperate म्हणजे wherever there is snow. Some part of India is tropical and some part is temperate. Temperate region is above Delhi or Himachal Pradesh, Jammu Kashmir. This is temperate region because there is high snowfall there. But tropical is Maharashtra is tropical. So tobacco area is maximum in Sangli, Kolhapur. There they are cultivating this tobacco. Then other parts of world, that is Brazil is there, Nigeria is there. Tropical regions they will cultivate this. So second important is if you are doing transformation here, then you will go for tropical species. In temperate region means USA, Europe, Russia, then Japan. These are temperate countries. There they will use Arabidopsis thaliana. This is the most model plant used for transformation. In bacteria it is E. coli. जसं E. coli आहे bacteria मध्ये तसं plant मध्ये Arabidopsis is there. So why they are using this Arabidopsis is because it has got 45 days life cycle. Within 45 days, the plant will grow, it will mature, it will flower, it will produce seeds, and it will die, and again next cycle will come. Means less than 2 months. In each year, you can produce 9 cycles. For tobacco, the life cycle is 3 to 4 months. मग ट्रान्सफॉर्मेशनला काय पाहिजे? लाईफ सायकल शुड बी शॉर्ट स्पॅन. Second importance is they have sequenced whole genome sequencing of tobacco is done. Arabidopsis is done. Tobacco is, they have not completed yet, but maximum they have sequenced it. And Arabidopsis also they have sequenced whole genome. So we know every gene is responsible for what in Arabidopsis. So if you are doing transformation, adding any genes, and you want to study the effect of that gene, what manipulations it is causing in the physiological processes in that plant, we can study in this Arabidopsis. Similarly in tobacco. So it is easy to handle. Life cycle is less. And Arabidopsis we are not using in India because it requires low temperature. But tobacco it requires high temperature. So it is used as a model plant for transformation. So why I have told everything about this tobacco is because if you want to do agrobacterium mediated transformation in tobacco, what you do, you excise the tobacco leaf. First you put the seeds, you grow it aseptically, then you will remove the leaf from that tobacco plant and you will put the disc. आणि हे डिस्क काय करणार तुम्ही, यू विल सस्पेंड दिस डिस्क इन द ॲग्रोबॅक्टेरियम सोल्युशन. ॲग्रोबॅक्टेरियम कल्चरमध्ये you will immerse this disk, and again what you will do, you will take this and you will put this on a ms media containing selection antibiotics. इथं काय होणार, from this leaf disk only you will get small plants, and these plants are transgenic. So this leaf regeneration method is used widely in tobacco. Got it? So here I have shown you the slide that is example of this tobacco, and from this tobacco small plants have emerged. कळालं सगळ्यांना?
Yes, sir.
See, I've shown you this tobacco. Same here it is done. See, they have excised the leaf in A figure. In B figure, they have put small disc 1 mm. This 1 mm disc they are suspended in agrobacterium solution and they have put it for culturing. Kyle mechanism, leaf culture, sir.
Yes, sir.
And after regeneration, they will go for rooting, hardening, and whole plant generation. Now, how one of this leaf culture seedling farming technique. So I will come to importance of this now culture. So how this technique has been used for in nursery. I'll give you example. In Tamil Nadu, a farmer he has developed a cost-saving, innovative leaf culture seedling farming technique, and he has got a lot of success in this. So what he did is, the man behind this is S. Ratnam. He is a 50-year-old farmer who lives in Tamil Nadu. Tamil Nadu is a South Indian state. Southern part of India it is present. He has got a bachelor's degree in applied science. He followed the path for agriculture. Even after he is educated, he is now 50 years old. So when he got bachelor's, he was around in 1980-85 he got his bachelor's. Then also he chose the path to do agriculture because of his heritage and passion for it. So he owns Eden Nursery Garden, India's first certified organic nursery, which is established in 1998 for helping farmers. And he is now commercially viable, and the nursery encourages the propagation of plant using leaf culture. Maximum they are using leaf culture. Currently, how much? Currently this nursery has production capacity of 15 to 20 lakh seedlings per year. This technique can be produced a wide range of plants, from flowering species to fruits. About the integration of this concept, he says what he says that he has executed this idea. He attended a 5-day tissue culture course in Tamil Nadu Agriculture University. See, we are aligned to Shivaji University, Kolhapur. So we have got science-based universities in India. So which are science-based universities? Pune University, Mumbai University, our Shivaji University, Nanded University. But agriculture colleges, they come under agriculture universities. In Maharashtra, we have got 4 agriculture universities at Rahuri, Dapoli, Parbhani. These universities are present. But in Tamil Nadu, there is a very one large agriculture university. Whole Tamil Nadu state, there is only one agriculture university. It is called TNAU, and it is very famous and very large university. Lot of research is going. So this farmer he attended a tissue culture course in Tamil Nadu, and he was fascinated by this simple method, and he introduced this into the leaf culture. So he did permutation combination and developed this leaf culture technique. So here you can see the photo of him, and backside are the plants which he has developed. Can everyone see?
Yes, sir.
So how does this help? This method is a cost saving. Here you don't require any laboratory. For example, you want to do callus culture, then you have to do explant, you have to remove, then you have to do aseptic sterilization, all those things. Nothing is required here. Here you will use hormones. He will remove the leaf, he will dip them into hormones, and he will put them into rooting mixture and put proper humidity conditions. And this leaf will develop into a shoot. So this method is a cost saving technique as it does not require seeds to grow crops. The leaves are available in abundance, thus making this method feasible. Besides, it maintains the genetic purity of the yield and promises assured produce to the farmer. Why this genetic purity is maintained? You're using vegetative method. Vegetative method मध्ये काय? Whatever the parent has got the characteristics, same characteristics the progeny will get. See, if you're cultivating vegetative rich plants, rose, then sugar cane, hibiscus, these are vegetatively. म्हणजे आपण काय करतो? Cut करतो stem and we put them into a soil and they will grow into shoots. But in sexual reproduction, what happens? Pollen grain comes and it develops on a stigma and then pollen tube is formed and this pollen male gamete it fuses with the egg cell. बरोबर ना? So parent मध्ये समजा हे असेल this female gamete is come from the egg part and male gamete the pollen grain may be from the same plant or different plant. मग इथं काय झालं? Permutation combination झालं. So there is probability that you will get new progeny. But here what happens? You are using leaf. So leaf is not any reproductive organ. बरोबर ना? Reproductive organ is flower and in this flower the seeds is formed. So here the genetic purity of and yield is maintained.
Yes, sir.
कळालं सगळ्यांना?
हो सर समजलं.
हाँ. So he did his first experiment with Jasminum, Sambac and Ixora species and he got success. And afterwards he devised this formulation for other fruit and leaf vegetable these varieties and he is producing this in a large manner. So this is the protocol he has used. See, here you can see in the pot the leaf is there. He has excised it. Then from this leaf what he has put it in the trays and from this leaf the shoots arise and from these shoots he has got whole plants. This is the tissue culture basic concept. Now how does this work? First what he does, he pluck a leaf and treat it with a tender coconut water for 30 minutes. काय केलं त्यांनी? First he removed the leaf and he has treated it with coconut water. Now coconut water is used in tissue culture. It is very beneficial in some plants tissue culture. This coconut water is used because coconut water will contain complex minerals, vitamins and amino acids and growth hormones. Then place the leaf petiole in a pocket filled with soil of tunnel mist chamber. Means what they did, they put a small shade net and their shade net in a tunnel form and the shade net ला वरती काय अटॅच केलं त्यांनी? Sprinklers and this sprinkle will sprinkle water in a mist form. म्हणजे काय केलं त्यांनी? See, the growth of plants is maximum in a rainy season. Why? Because environmental moisture is very high. So he has control the environment and artificially he has developed rainy like condition. Mist तयार केले. Then second, cover the leaves with airtight polythene to maintain the moisture. Polythene the leaves ला cover केलं. Then place the chamber in an 8 mm frame and remove the polythene every 5 days to check the moisture level. Polythene काढायचं दर 5 दिवसांनी आणि spray the water accordingly. Third is the procedure takes around 4 to 5 weeks depending on the species. Once the sapling is ready, it can be planted on the farm for growth. The farmer is making efforts to expand the scope of this work and teach farmers to adopt this. He is taking many workshops to teach this cost effective method to farmers. So see, one man, he started with a mission and he has developed this technique. How extensively? Same here they have showed. Now I'll show you how he has put this leaf. See, here is this small bags are there. He has put this cocoa pit and all this mixture, and in that mixture he has dipped this leaf and put it for culturing. दिसलं सगळ्यांना?
Yes, sir. Yes, sir.
ठीक आहे. So next see how, see this after 3 to 4 weeks also the leaves are healthy. का dead झालेत नाही. And see he has uprooted these leaves, and from this leaf he can chew the shoots. दिसलं सगळ्यांना? How shoots and roots have developed from a leaf.
Yes, sir.
See the mature plant. So no one can believe also that these plants have been developed from leaf. कळालं सगळ्यांना ही मेथड?
हो सर समजली.
Now last is importance of the leaf culture. So now culture of excised leaf primordia is valuable to study the effect of nutrients, growth factors, and changing environmental condition on leaf development under conditions divorced from the complexities of the intact plant. Now intact plant मधनं काय केलं त्यांनी? They have isolated the leaf, आणि ह्या लीफला काय केलं? व्हेरियस न्यूट्रिएंट आणि ग्रोथ कंडिशन्स मध्ये स्टडी केलं. then what you can know, for example you are putting this leaf into the saline water, what will happen? Chlorophyll synthesis will be hampered. If the salinity is high, that is toxic to the plant. If you are putting this leaf into drought conditions, if it is drought resistance, the leaf will grow normally. Then you are using gibberellic acid, other hormones, मग काय changes होतात? So in vitro studies this leaf culture is very important. In case of leaf primordia, firm leaf primordia, cultures are used to study the formation of sporangia and the size at which the primordia is destined to become a leaf. Now या स्पोरॅन्जिया मध्ये काय असतात? तर रिप्रोडक्टिव्ह ऑर्गन्स असतात. तर त्या रिप्रोडक्टिव्ह ऑर्गनच्या स्टडीसाठी फर्म मध्ये हे वापरलं जातं लीफ कल्चर. Then young leaf of most of the Solanaceae species. Solanaceae means whatever you are using, brinjal, tomato, and potato. These contain to Solanaceae species. From numerous shoot buds instead of callus when they are cultured in solid MS medium. Means if you are culturing the leaf without forming callus, they will produce numerous small shoots. So they will use this method to produce the plantlets. These are importance of the leaf culture. Thank you. तो कळालं सगळ्यांना?
हो सर कळालं.
तो काय डाऊट्स आहेत का?
नो नो सर.
बाकीच्यांना काय ऐकू येतं का आवाज, का फक्त फिजिकली युअर प्रेझेंट? अनिकेत
हां सर.
कळाली कन्सेप्ट?
हो सर.
सो विल स्टॉप इट दिस. आपण सकाळचं सीओसीचं कंटिन्यू करायचं लेक्चर.
हो सर चालेल कंटिन्यू केलं तरी.